Welcome back to Falcon Physician Review's online review for USMLE Step 1. This is Microbiology Module 37. We're going to talk about retroviruses. This is a long module. It's mostly going to focus on HIV, or human immunodeficiency virus. We'll talk about how the virus works, its replicative strategy, how it establishes a latent infection, which cells in the immune system it targets at first, and then later how it targets the CD4 positive T cell, and how that brings about our ultimate destruction of our immune system. That brings about immunodeficiency and sets us up for opportunistic infections from other diseases. We'll go through each of these at a time. There'll be lots of points on this for Step 1, so you should know the material cold. We'll go through it and try to explain it in a way that'll make sense. Welcome to Falcon Physician Review's online review for USMLE Step 1. This is Microbiology, Module 37. We're going to talk about retroviruses. Retroviruses come in two groups that are clinically significant. The lentivirus group, which has human immunodeficiency viruses 1 and 2, and the oncogenic retrovirus group, HTLV virus 1 and 2. We're going to talk about HIV and AIDS first. Historically, HIV was first discovered in the 60s and 70s when there was a number of pneumocystic coronary pneumonias that showed up in Los Angeles. In 1979, there were 5 cases of PCP. They were all homosexuals. Then we had GIM sustained at high magnification, which shows a faintish blue dot in this picture. That shows the PCP lesions in this cytologic preparation from bronchoalveolar lavage. Then, in early 1981, you had 5 cases of Kaposi sarcoma. Why? This is a rare disease and only seen in immunocompromised patients. In 1981, there were 26 cases of Kaposi sarcoma. They were all found in young men in San Francisco and New York, and they were all homosexuals. So there was a pattern existing that showed up of people that were homosexual, Haitian, heroin addicts, or hemophiliacs. All these people had these immunocompetent diseases. Risk factors for HIV include promiscuous sex, prostitutes, homosexual or bisexual men. Also, intravenous drug use, female sex partners of HIV-positive IV drug users, blood transfusions, hemophilia, or vertical transmission. AIDS is defined as HIV infection plus a CD4 count of less than 200 cells per microliter of blood. This leads to opportunistic infections, secondary neoplasms, and neurologic manifestations. In order to have AIDS, you must have 2 or more AIDS-defining conditions, including fungal infections like candidiasis, cancers like Kaposi sarcoma, Burkitt's lymphoma, primary CNS lymphoma. You must have certain viral infections like CMV retinitis, or herpes simplex, chronic ulcers, or pneumonitis. You must have parasitic infections like toxoplasma of the brain, or cryptosporidiosis, or chronic intestinal inflammation. You must have a bacterial infection like Mycobacterium tuberculosis at any site, or the Mycobacterium avium complex. Disease progression starts initially as a mono-like disease with viremia. You get infection via the CCR5 cells with myeloid and T cells. Then you have a decrease slowly of the CD4 positive cells. The virus switches to infect CCR5 and CXCR4 lymphoid cells. Then you get mild opportunistic diseases. As you lose all your CD4 cells, the next ones that go are the CD8 cells, and then you get severe opportunistic diseases. This figure is important because it defines what happens with your CD4 cells and with your virus levels over time. Initially, within the first few weeks, you have a high spike of virus antigen, which is your acute infection. Then the immune system takes over. You get antibody that shows up in the blue line, and your CD4 cells slowly go down before you get any problems. That's a chronic lymphadenopathy stage. A subclinical immune dysfunction stage happens as your T cells go lower and lower, but you still don't have any positive virus shown. Once you get skin and mucous membrane immune defects, then the virus starts to show up, your antibodies drop, and your CD4 levels are well below what you need to keep you alive. Then you have systemic immune deficiency, opportunistic infections, and often death. Similar situation here in this figure with HIV and AIDS, the cellular and immunological picture and the course of the disease. You have CD8 cells that stay up high until the very end, and as they drop, you get AIDS. The CD4 cells drop early and can be over a prolonged period of time, but once they get down below 200 or so, you start to get in trouble. and then as you lose suppression, the viral antigen peaks up again. Acute infection with HIV is associated with high virus titer and mild symptoms. It's like mono. You get a fall in the CD4 positive T cells, but recovery. You get a rise in the CD8 positive T cells. And you have a high virus titer, up to 10 million viruses per ml of blood. The macrophages are initially infected by a CCR5 coreceptor. They are the gatekeepers of infection, and they're resistant to the cytopathic effects of HIV. Through this phase, you get widespread dissemination and seeding of the lymphoid organs. The next step is a strong immune response, where the virus disappears from circulation. You have a good cytotoxic T cell response, and you have soluble antibodies which appear later, both against surface and internal proteins. Most of the virus at this stage comes from recently activated or dividing infected CD4 cells. The CD4 positive cell production compensates for the dying CD4 cells, and so you're able to maintain those lines. You've got loss due to lysis of cells by virus production, and you've got destruction of cells by cytotoxic T cell lymphocytes. Then you enter the latent stage, where you have latent virus production and latent symptoms. You get upregulation of viral proteins such as TAT and REV. NEF is a viral protein which downregulates the virus. The virus can persist in extravascular tissues, like lymph nodes with dendritic cells. You've got a resting CD4 positive memory cell that lasts a very long time, and this is a stable population of cells which hold the virus while everything is latent, waiting for the reactivation stage. When you get the beginning of disease is when you have a massive loss of CD4 positive T cells. They're the targets of the virus. The cells that proliferate to respond to the virus are killed by it, so you're killing your own immune system. Dendritic cells present antigen to the virus to CD4 T cells just as they're activated. Then you get mutation of the virus during reinfection. You change your epitopes in your viral proteins to escape from immune response. You get antiviral drug resistance, and you get death of the CD4 positive T cells. Now we've entered into the advanced stage where you have AIDS. CD8 positive cells are destroyed by more CD4 positive cells. The CD4 cell loss means virus and infected cells are no longer controlled. Your CD4 positive cells fall below 200, and the virus titer rises rapidly, and your remaining immune response collapses. Then you get death of the CD8 positive cells, opportunistic infections, and death within 2 years without intervention. Questions. Which of the following regarding the immunologic picture of acute HIV infection is false? A. High viral titers. That's true. B. Slight drop of CD4 followed by recovery. That's true. Drop in CD8 count? No, you don't see that till the very end where you march into AIDS. D. Detectable anti-GP120 antibody? Yeah, you'll see that in acute infection. Next question. The end of the acute phase of HIV infection is manifested by which of the following? A. Drop in detectable HIV viral titer of the P24 antigen level? That's true. B. Recovery of the CD4 positive cell count? That's true. Rise in the HIV-specific CD8 positive T cell response? That's true. So D, all of the above is correct. This figure shows the correlation between HIV and AIDS. There is a good correlation between the number of HIV genomes measured by PCR and the progression to disease. So you can see the more HIV RNA genomes you have, the more likely you are to have AIDS. This next figure also shows that viral load predicts survival time. So the relation between the baseline viral load and the median survival time. So the higher your initial viral load, the less time you have to live. Because the more virus you get involved, the more CD4 T cells get infected, the less time you have to live. Let's talk about HIV, the virus. The membrane is host-derived, and it has two viral glycoproteins, which is GP160, which can be cleaved to GP41 and GP120. You'll find these antigens in the blood. There is the ENV gene, which is the envelope gene which encodes these, and GP41 mediates fusion. There are group-specific antigens that these sequences code for proteins inside the envelope, and these are the major structural proteins that are antigenic. We've got P17, P24, which codes for the capsid, and P7 and 9, which are associated with the RNA. These come from the GAG gene. Enzymes involved in the virus include reverse transcriptase, which produces a double-stranded DNA provirus. Integrase, which helps the proviral DNA integrate into the host cell, and protease, which cleaves the polyproteins during the assembly process. These are encoded by the pol gene for polymerase. GP120 binds to CD4 and CCR5, which is found in the macrophage, dendritic cell, and activated T cell line. Later in the stage, it develops a CXCR4 tropism, and other T cells are involved with that co-receptor. There's membrane fusion involving GP41, which allows the nucleocapsid entry, followed by release of the genomic RNA. The viral reverse transcriptase then makes a double-stranded DNA copy, which migrates to the nucleus. Integrase helps it get the viral DNA into the human chromosome. And HIV proteins regulate transcription, RNA cleavage, and RNA transports to ribosomes and protein cleavage. The genome of HIV has a number of the genes we've already talked about, like GAG, POL, ENV. You've also got TAT and VIP, and you've got long terminal repeats on each side. So it has 9749 nucleotides, 3 structural genes, long terminal repeats, and extra open reading frames that are due to latency. These open reading frames code for 15 small proteins and antibodies in AIDS patients. The long terminal repeats serve 2 important functions. First off, they have sticky ends, which are sequences which are recognized by the integrase that are involved in the insertion of the host DNA. And second off, they have promoter or enhancer function. Once they're incorporated into the host DNA, proteins that bind the long terminal repeats can modify viral DNA and have DNA transcription. Structural components of the HIV genome include small, non-structural proteins early, like TAT, which is a transcriptional activator of translation, REV, which is a regulator of virion protein expression, and these are essential for HIV replication. NEF is interesting. It's a negative regulatory factor, and those are the early genes. Later, you have VIF, VPU, and VPR, which are viral infectivity proteins. The progression to disease slowed significantly in the presence of NEF because it negatively regulates the genes. Promotion downregulates CD4 and MHC1. Questions. Antibodies against which component of HIV neutralize infectivity? So if we're going to neutralize infectivity, we've got to get antibodies against a cell surface protein. This is not going to be P24 or P17 or capsid. Those are all inside the envelope. But GP120 is a cell surface marker. So if you've got antibodies to that, you're protected. Which of the following is regarding the ENV gene of HIV is true? A. It encodes viral lipoproteins. B. It encodes viral hemagglutinins. C. It's highly variable. And D. It's very stable. Well, actually, it's C for highly variable. The ENV gene encodes the protein GP120, which is on the surface. It doesn't encode viral hemagglutinins, and its instability helps the virus evade the immune response. Next question. A 26-year-old sexually active male presents with weight loss, lymphadenopathy, and night sweats. His blood counts are remarkable for CD4 counts less than 500. Which of the following is the most likely offending pathogen? So we have somebody who's immunocompromised, and they've got weight loss, lymphadenopathy, and night sweats. So you worry about somebody who has acute HIV, which is going to be an enveloped, diploid, single-stranded mRNA with positive polarity. Next question. A 26-year-old male has lost 15 pounds during the last 3 months and complains of fever, chills, cough. He's found to have multiple infections including tuberculosis, oral candida. His CD4 count is 50. Which viral protein may be important in causing the patient's disease, but not essential for replication? So we think about genes that are essential for replication. You think about GAG, you think about GP120, and you think about integrase. Without those, they're not going to be able to replicate. NEF is not essential for replication, but is important in causing the disease. This picture shows the replication cycle. We have surface GP120 binding to the CD4 positive T helper cells, macrophages, and microglia. HIV is then taken into the cell, it loses its envelope, and the RNA is uncoded. The RNA is copied using the virion-associated reverse transcriptase. It ultimately leads to double-stranded DNA with long terminal repeats. The DNA then migrates to the nucleus, where integrase helps it get into the host DNA, forming the provirus. There, TAT can upregulate transcription, and REG transports the mRNAs to the cytoplasm. Transcription produces some single-stranded positive RNAs, some spliced and some intact. The spliced RNAs will be used as mRNA, The whole RNA is used as genomic RNA. Translation then produces the proteins, some of which are polyproteins, which are cleaved by the HIV protease. You then have assembly of the entire virus and maturation and release. Question: why would a long-term non-progressor of HIV be able to escape AIDS? A. They never become ill from HIV. Well, they have HIV, and so they've got it in their system. B. The immune system eliminates the virus from the person. That would be one way to do it, but we haven't been able to see that humanly possible yet. C. A defect in the NEF protein decreases the virulence of the virus. That actually would work. We know that NEF regulates decreases the infectivity of the virus. D. The virus infects macrophages and T cells. That would lead to AIDS. Or E. The GP120 interacts with CXCR4. That would also lead to AIDS. Chemokine receptors in HIV tropism. Initially, the virus is M-tropic. It affects macrophages, and these strains affect monocytes and macrophages in the peripheral blood. They use CCR5. Then it switches to be a T-tropic strain, which infects T cells, using CXCR4. Acute infection, like I said, is M-tropic, and it evolves to T-tropic strains owing to mutations in GP120. T-tropic strains accumulate, and they herald the final rapid phase of disease progression. This figure shows that GP120 binds to CXCR4 or CD4 and uses co-receptors to enable membrane fusion. The co-receptors initially are CCR5, and later, as it turns to T-tropic, are CXCR4. Macrophages are known as the Trojan horse. They form a reservoir outside the blood for HIV to replicate and change to be T-tropic. They carry the virus into different organs, such as the brain. They're non-proliferating, mature macrophages, and they sustain HIV production for a long time without being killed. Dendritic cells use GP120 interaction. HIV GP120 binds to macrophages or dendritic cells with CD4. The virus is opsonized by anti-GP120 antibodies, which binds to the macrophages and the Fc receptor. Question: which of the following statements about the interaction of HIV with macrophages is correct? A. HIV never infects macrophages of the brain or spinal cord. Well, that's not correct. We know that it does. B. Pulmonary macrophages are never infected with HIV. Be wary of statements that say never. Pulmonary macrophages can be infected by HIV. B. The infection of macrophages is unrestricted and continuously active. Well, that's not true because we know that they can hold latent disease. And D. Macrophages are resistant to the cytopathic effects of HIV. That's true. Next. Which of the following host factors has been associated with increased resistance to HIV infections? A. Mutations in CCR5. That's true. If you have mutations in CCR5, then you'll be resistant to HIV infection because you can't get the initial target cell, the M-tropic virus. Dynamics of CD4 positive T cells in an HIV infection. So this picture talks about what happens with HIV T cells. If you have an uninfected activated T cell, it can go on to become infected, in which it returns to its resting state and is long-lived, holding the virus. That's the chronically infected memory T cells with provirus DNA. We don't like that very much. Also, we don't like it when cell death or immune destruction kill our infected T cells. We need to get rid of the infected cells, but when we get rid of our immune system, that causes the problem. So finally, we have this smaller and smaller pool of unactivated, uninfected memory T cells, which eventually get involved, and we lose all of our T cells and march on to AIDS. The inexorable decline of CD4 positive T cells is described as we did before. The question is, as we see this march, we keep losing more and more T cells as they become infected and killed by the CD8 positive C cells. So CD4 positive T cells disappear because they can be lysed by the virus, they can be killed as in syncytia, or they can be killed by CD8 positive T cells. Virus destroys the cell as a result of budding, but few of the cells are infected. In the early stage of infection, only 1 in 10,000 cells are infected, but in the late stage it's much more potent, 1 in 40. HIV could kill an entire subpopulation of precursor cells, and people develop AIDS when they have HIV that does not lyse cells. CD4 cells disappear as they become killed by CD8 positive T cells. The syncytia are not very common, and when they do form, they're very fragile. Most T helper cells are not HIV positive, and they could sweep up uninfected cells, and the syncytium may be poor or ineffective at immune response. The third way is through the cytotoxic T cell killing HIV-infected CD4 positive T cells. The killing of the CD4 positive T cells through complement-mediated lysis is the last way which we get rid of our population and march towards AIDS. Population polymorphism is important. Reverse transcriptase produces complementary DNA for the integrated genome, and the retroviruses use the host RNA polymerase II to replicate the integrated genome. Reverse transcriptase and polymerase II have a high error rate, and that's why we get so much variation. The HIV genome has less than 10,000 nucleotides, and therefore every new virus has at least one mutation. This means that the virus that you get originally is much, much different than the virus that you get when AIDS appears. The antigens change, and the drug sensitivities change. Vaccine problems come because HIV envelope glycoproteins mutate rapidly, especially the V3 loop of GP120. It's called the hypervariable loop. And antibodies against this neutralize the infection initially, but then they're rapidly changing, and so you don't get the same quasi-species in the same person. We need to target conserved regions like the binding CD4 site. Question. The rapid replication of HIV coupled with genetic variability leads to many variants of HIV in a single infected patient in the course of an infection. Thus, HIV can rapidly develop resistance to antiviral drugs. This variability in HIV is caused by A. Gene conversion, B. Gene duplication, C. Hypermutability, or D. None of the above. Well, the answer is C. It's hypermutability or hypervariability. Which of the following HIV viral components is most susceptible to mutations, resulting in the formation of quasi-species in an infected individual? A. The hypervariable region of GP120. That's what it is. That's the thing that causes the most difference in the HIV quasi-species. Anti-HIV strategies include HART, or highly active antiretroviral therapy, which is 2 nucleoside analogs, reverse transcriptase inhibitors, and 1 protease inhibitor. We now also have 2 nucleoside analog reverse transcriptase inhibitors and 1 non-nucleoside analog reverse transcriptase inhibitor. This table shows the various anti-HIV drugs. Basically, you should be familiar with which drug fits in which category. And as you know, we have nucleoside analogs, protease inhibitors, receptor or fusion inhibitors, and non-nucleoside reverse transcriptase inhibitors. Prevention can happen by blood and organ screening for HIV. This is something that we've recently been able to do, and so our populations of hemophiliacs who have died from HIV have dropped significantly. Education about sexually transmitted disease, safe sex, new needles, and taking your meds religiously can also prevent infection. Programs such as needle exchanges and condoms can also help. Education did lead to a leveling off of the rate of the increase in AIDS, but HART has greatly slowed the death rate. The fact that fewer people are dying per year from the infection means that the number of HIV-infected people in the population is actually rising, even though we have less new cases. Unless education continues to be successful, and unless we can cure infected people of the virus, the problem of the virus spread is and will continue to be with us. This slide is important because it lists the opportunistic organisms that happen at each CD4 level. So you should be familiar with what happens with your CD4 count as they get less than 500, greater than 500, greater than 200, and then less than 200, 150. CMV retinitis is the hallmark as you get CD4 counts less than 50. Other opportunistic infections are listed, and they're less commonly encountered. Management changes depending on the CD4 positive cell count. If you have less than 500 or a detectable viral load, you need to have HART, highly active antiretroviral therapy. If your CD4 count drops below 200, you should start a Bactrim prophylaxis to prevent PCP or toxoplasma. As it gets less than 100, people should be on azithromycin as well to keep you from getting Mycobacterium avium intracellulare infections. Diagnosis of HIV happens by a combination of an ELISA test and a Western blot. You use PCR for neonatal infections. When you monitor patients, you want to monitor their compliance and their therapy. You check the viral load by PCR to amplify the genome, and you look for other genome quantification. You always want to know the CD4 count because that tells you where they are in terms of opportunistic infections. Question. Which of the following tests is ideal for monitoring in HIV-positive patients' progression to AIDS? A. Nucleic acid amplification test for HIV RNA. That's a pretty darn good one. If you know how much HIV they have, you know how quickly they're progressing. The HIV P24 antigen is often negative until it's very late. HIV antibodies can be variable, and the CD4 lymphocyte count is most used for clinical management, but it doesn't tell us the progression towards AIDS. This is a picture of Kaposi's sarcoma, both clinically and histopathologically. This is a picture of a ring-enhancing lesion in a patient with HIV and toxoplasmosis. This is a picture of oral candidiasis or oral thrush. Question. A 21-year-old female with AIDS develops left-sided weakness involving the upper but not the lower limb. MRI scan reveals a ring-enhancing lesion within the white matter of the left frontal lobe. Biopsy shows coagulative necrosis of brain parenchyma with inflammatory infiltrate admixed with microscopic cysts that contain characteristic bradyzoites. Which of the following is the most common source of this type of infection? So we're talking about Toxoplasma gondii, a frequent CNS infection in HIV people, and we get it from cats. This is a picture of oral candidiasis or oral thrush. This is a picture of cytoplasmic effects from pneumocystis. Question. A 27-year-old male presents with fever, night sweats, cough, and weight loss. He has multiple sex partners. His chest x-ray shows bilateral pulmonary infiltrates with interstitial and alveolar markings. His CD4 count is 125. The most likely diagnosis? So this is one of the diagnoses that initially led to the discovery of HIV. CD4 count of 125 with interstitial alveolar markings is going to be pneumocystis pneumonia. Pneumococcal and staphylococcal pneumonia are going to give you more of a lobar consolidation. And your risk factors are not going to be for influenza so much as they are going to be for HIV and PCP. The least invasive and least expensive diagnostic approach to diagnose pneumocystis pneumonia is sputum induction using aerosolized saline. A conventional sputum sample is going to be difficult because it doesn't produce as much mucus as the bacterial pneumonias. Bronchiolar lavage is good, but it's invasive, and so is an open lung biopsy. Last, we have a synopsis of classic viral manifestations, which is kind of a quick checklist for high yield, as you'll see on USMLE Step 1. If you see a rash with a slap cheek appearance, you should think of fifth disease and parvovirus B19. If you have a descending maculopapular rash and Koplik spots, you could think of measles. If you have gastroenteritis followed by paralysis, you think of poliovirus infection. Cervical cancer in a sexually active smoker, you think of HPV, serotypes 16 and 18. Parotitis, orchitis, and possible maral sterility, you think of mumps. Cataracts with blindness in newborns, think of rubella. If you have a vesicular lesion in a dermatomal pattern, you've got activation of varicella zoster. Acute retinitis in a patient with acquired immunodeficiency, you should think of CMV retinitis. Genital warts are from HPV, serotype 6 and 11. Painful genital vesicular lesions are HSV2, but sometimes HSV1. High mortality hepatitis E in pregnant women, you think about hepatitis E. Encephalitis following an animal bite with intracytoplasmic Negre bodies on neurons, you think of rabies. If you have a teenager who's tired, has a big spleen, atypical lymphocytosis, and positive heterophile antibody tests, that's mononucleosis from EBV. You're on a cruise ship and you've got gastroenteritis, think of Norwalk virus. You're in a daycare and you've got gastroenteritis in your child, think of rotavirus. You've got the common cold, think of coronavirus or rhinovirus. Bronchiolitis in children is most likely going to be RSV. And neonatal encephalitis, you think of herpes simplex or cytomegalovirus. Question. A patient develops fever, shortness of breath, and appears to be quite ill. X-ray demonstrates bilateral interstitial lung infiltrates. Bronchial washings demonstrate a small hat-shaped organism, visible on silver strain within the alveoli. His CD4 count is 50. Which predisposing condition is most likely to be present in this patient? This is another question of pneumocystis carinii, pneumonia, and we all know that with a CD4 count of 50, the patient has AIDS. Other retroviruses include endogenous retroviruses and oncogenic retroviruses. There are no human fast oncogene viruses, but HTLV is a slow, stimulating growth by other means virus. HTLV binds to CD4. It stimulates T cell growth by upregulating IL-2 and IL-2 receptors. It takes about 30 years. And it causes acute T cell lymphocytic leukemia, ATLL. You'll see HTLV common in regions of Japan, the Caribbean, and Central Africa. It's not very common in the United States. It is transmitted like HIV. The diagnosis depends on the presence of flower cells or lobulated nuclei, Sézary syndrome with skin-like lesions. RT-PCR will diagnose the viral RNA, and you can use an ELISA to detect the antigens. Wrapping up Module 37, we spent a lot of time talking about HIV. We also talked about HTLV, the other clinically important retrovirus. Retroviruses have their own reverse transcriptase, and we have antiviral medications which target that. Since the advent of highly active antiretroviral therapy, the disease burden of AIDS has actually been greatly improved. We have people who have HIV, but they're living much longer, and they're not dying of AIDS complications. You should be very familiar with the presentation of AIDS. You should also be familiar with some of the major components of the viral genome. How the disease starts by activating our macrophages and then switching to our CD4 positive T cells. The selective destruction of this cell population leads to our immune dysregulation and sets us up for opportunistic infections. You need to be familiar with the different classifications of immunodeficiency based on your CD4 positive T cell count and which opportunistic infections take place at which level. You should be familiar with CMV retinitis as it's commonly found in AIDS patients. We're going to go next to the next module, 38, and talk about mycology, or fungal growth.